दोस्तों, मानता हूँ ये हमारा बुरा वक्त चल रहा है। वीडियो में मैं कोरोना वायरस का खुलासा करने वाला हूँ। को कोविड-19 या सार्स-कोव-2 भी बोलते हैं। ये हमारे शरीर को बहुत बुरी तरह से प्रभावित करता है। ये आरएनए प्रोटोटाइप जेनेटिकल वायरस है। इसको बायोलॉजिकल वेपन का दर्जा भी दिया गया है। ये बेसिकली छुआछूत की बीमारी है। अभी तक ये पता नहीं लगा है कि ये कोरोना वायरस कहाँ से आया है। कोरोना वायरस का असली चित्र आपके सामने है, जो कि मुकुटाकार का होता है। इसमें 3 तरह के प्रोटीन रिसेप्टर होते हैं और इंसानों के शरीर में फैलने के लिए ज़्यादा समय भी नहीं लेता। ऐसे वक्त पे बहुत से लोग अपना योगदान दे रहे हैं और देश की भलाई भी कर रहे हैं। लेकिन बात यहीं पे ख़त्म नहीं होती, क्योंकि ये इंसानी शरीर में बहुत तेज फैलता है। कोरोना वायरस के चलते इंसान के शरीर में बहुत सारा कोरोना वायरस बनने लगता है। जब इंसान छींकता या खांसता है तो ये कोरोना वायरस बाहर आ जाता है और हवा के सहारे किसी दूसरे इंसान के अंदर चला जाता है। असली काम गले में शुरू होता है, जब ये गले में मौजूद कोशिकाओं में मौजूद रिसेप्टर से बाइंड करके सेल मेम्ब्रेन के अंदर चला जाता है।
A young man that had no comorbidities at all. And so that's actually rather concerning. So what about treatment? Well, as it turns out, Wuhan. So what should you do? Well, I would have...
The large S glycoproteins are used by the virus to gain entry to human cells. They attach to receptors on the cell membrane. This binding convinces the cell that the viron is not a threat, allowing the virus entry. The exact mechanism for this is not known. Possibly the virus binds with the human cell's membrane, releasing its contents into the cytoplasm. Alternatively, as shown here, the human cell ingests the virus in a process known as endocytosis. Once inside the cytoplasm, the endosome opens to reveal the virus's genetic material, a single-stranded RNA. The virus hijacks the cell's machinery to replicate the RNA and N proteins, and uses the endoplasmic reticulum to form its M protein outer layer, and the all-important S protein. After replication, the virus is carried by the Golgi bodies out of the cell in a process known as exocytosis, so that it can infect other cells. Meanwhile, the stress of viral production on the endoplasmic reticulum eventually leads to apoptosis, or cell death. It should be noted that the mechanism of action for 2019 novel coronavirus is unknown. However, scientists in China have sequenced the genome, while others have used the sequencing to visualize the structure.
So coronavirus is a group of viruses, a family of viruses. They have their big RNA viruses. They have actually some of the biggest RNA genomes of any virus.
19 novel coronavirus is a newly discovered strain from the coronavirus family that causes respiratory disease, PARTS. Inside the virus lies the genetic encoding that allows the virus to hijack human cells and turn them into virus factories. A protein encapsulates the genetic material known as the viral envelope. On the surface of the viron are S and HE proteins. The structure of 2019 novel coronavirus is a mutation. For more info on this, see the link below. The large S glycoproteins are used by the virus to gain entry to human cells. They attach to receptors on the cell membrane. This binding convinces the cell that the viron is not a threat, allowing the virus entry. The exact mechanism for this is not known. Possibly the virus binds with the human cell's membrane. Alternatively, as shown here, the human cell ingests the virus in a process known as endocytosis. Once inside the cytoplasm, the endosome opens to reveal the virus's genetic material.
What are viruses? Of the many things that can make you sick by infecting and reproducing inside you, viruses are among them. Viruses are a huge spikes sticking out of their membranes. These protein spikes are selectively sticky, sort of like Velcro. They don't attach to most objects but are extremely sticky when they bump into specific molecules found on the outsides of animal cells. Once held firmly, when virus genes are being copied during reproduction, mutations can occur. These are either due to simple copying errors or processes called reassortment and recombination. These happen when two or more viruses infect a single cell. In most cases, mutations that change the shape of viral spikes render the virus useless. Their Velcro no longer sticks to any host cells. On rare occasions, however, a chance mutation will just happen to allow a virus to attach to a new host species. If the modified virus is then lucky enough to encounter that new host species, infection can occur. Also make it worse at infecting its original host. Because of this, many spillover viruses go extinct after infecting just one or two people. They're usually dead ends. That said, if the virus can survive and reproduce just long enough, natural selection will promote any new mutations that help it better spread and reproduce in the new host population. Positive mutations accumulate over multiple generations, but there are over 7.5
estimated 50 to 100 million deaths. While no global pandemic has been born from the family of coronavirus, two regional epidemics have resulted from this family of viruses. They include severe acute respiratory syndrome, or SARS, and Middle East respiratory syndrome, or MERS, in 2001 to 2003 and 2012 to 2015, respectively, and resulted in are sick. Pandemic risk of the novel coronavirus is very real. However, biology has advanced greatly in the 15 years since SARS, and so has communication between biologists and governments. China has taken monumental